Okay, folks, welcome back to Respiration Part 2. Remember, this is Chapter 9 that we're finishing up. And so let's just review what we discussed in great detail. In the first part of the PowerPoint, we were looking at aerobic cellular respiration that occurs in the cytoplasm and the mitochondria of eukaryotic cells. And the end result of aerobic cellular respiration was 32 ATP. Now we're going to look at something slightly different, and this is referred to as fermentation. And fermentation is a type of anaerobic cellular respiration. It does not use oxygen or the electron transport chain. And it is used by bacteria in marshes, sewage treatment plants, and the making of yogurt. And in this, the final electron acceptor is an organic substance instead of oxygen. This starts with the process that we discussed in the last unit called glycolysis. And remember, the glycolysis takes place in the cytoplasm of the cell. And that process is where glucose is converted to 2-pyruvic acid molecules. And the end result of that is 2-NADH, remember they are electron carriers, and 2-ATP. So we're going to discuss two different types of fermentation. The first type that we're going to discuss is called lactate fermentation. And it is used commercially in the making of cheese, yogurt, and sauerkraut. And in this type of fermentation, the 2-pyruvic acids that were produced during glycolysis ultimately get converted to 2 lactate molecules, hence the name. This type of fermentation is also used inside our bodies when oxygen is not available. So if our cells don't have oxygen readily available, our cells have the ability to undertake lactate fermentation. However, this can't last very long. It is used by us for short bursts of speed. If somebody were to walk in the room and start chasing you right now, your body would be using lactate fermentation. However, if they kept chasing you, you would have to start breathing heavily and your cells would have to convert to aerobic respiration. So when glucose is gone, the muscles cannot contract. So muscles can only use this when oxygen is in short supply. And again, it doesn't last for a long period of time. And so there is a similar picture to this on page 180 of your books. So we start out with that glucose, just like we talked in the previous unit, and it undergoes glycolysis. And that glycolysis takes that 6-molecule or 6-carbon glucose and converts it into 2 3-carbon molecules of pyruvic acid. That process produces 2-NADH and 2-ATP. Okay, and we ultimately end up with 2 lactic acid molecules. And those lactic acid molecules are also 3-carbon each. And so here's the simplified version of that. And just to give you an idea, we're not going to get into the phosphocreatine system, but here's just an idea if you were a biker on a long bike ride, how your body would convert. So it's using both at the same time, but if you're pack riding, you're far above the anaerobic respiration, you're using mostly aerobic respiration. You're breathing hard, your cells are getting enough oxygen. If you hit a hill all of a sudden, then not all of your cells are going to have oxygen available, so your anaerobic respiration will increase. And then finally, for your final sprint, you'll have a fair amount of anaerobic, but again, mostly aerobic. So you can see here that your body's undergoing both kind of all the time. And this is just a different graphic showing kind of how a marathon runner they would be in primarily aerobic respiration, obviously with lots of training. The swimmer could do the lactic acid system for about a minute and a half. And then the sprinter could use the phosphagen system. That's the phosphocreatine system we're not going to discuss. But that just gives you an idea. So the whole point of training for a marathon is you just don't go out and run a marathon one day. If you do, you're going to be hurting. But you have to train for that. And the point of that is to train your body to stay in aerobic respiration for longer periods of time. So once your body starts losing that oxygen, then it's going to convert to the lactic acid system of fermentation, and you're going to feel very tired. And part of the reason for that is you're only getting 2 ATP for every molecule of glucose. Whereas if you keep getting oxygen to your tissues, then you can get those 32 ATP for every molecule of glucose. So let's talk about the second type of fermentation, a type of fermentation that is near and dear to all college students' hearts, and that is alcoholic fermentation. And that is not only used in the making of beer and wine, but it is also used by yeast cells in the making of bread. And in this case, the 2-pyruvic acids are converted to 2-ethanol, which is a type of alcohol. During this process in the making of bread, the yeast releases carbon dioxide during this process, and that the release of those bubbles of carbon dioxide is what makes the bread dough rise. So when you look at your slice of bread, you can see all those little nooks and crannies. That is from the release of carbon dioxide during alcoholic fermentation. And here's the process. Again, we start out with glycolysis, and that has a net yield of 2 ATP and NADH, and then we convert that 1 molecule of 6-carbon glucose to 2 molecules of 3-carbon pyruvic acid, and then eventually those get converted to 2 2-carbon molecules of ethyl alcohol or ethanol. And in that process, to follow the carbons, there are 2 molecules of carbon dioxide released. And again, there's a similar picture to this on page 180 of your books. And so here's the very simplified process. And again, they don't show it, but there'd be 2 molecules of carbon dioxide being released in this process as well, to make sure that we account for all 6 carbons. And so this process is used in the production of beer. If you've ever been to a brewery, you will notice that the containers that they are using don't have open tops. They are closed, and that is to keep the process anaerobic, to keep oxygen out. So let's talk about the type of organisms that undergo these different types of processes. We have what we call facultative anaerobic organisms, and they can use aerobic or fermentation respiration. So our muscle cells are facultative anaerobic cells. So when enough oxygen is present, they use aerobic respiration and produce how many ATP? That's correct, 32. When there is not enough oxygen present and they use lactic acid fermentation, how many ATP do they produce? 2. So just to give you an example of what happens in our muscle cells, that lactic acid is later pumped to the liver, where it is converted back to pyruvate and used in the rest of the aerobic respiration cycle to produce the remaining 30 ATP. How do we get oxygen to our muscle cells? We breathe. We breathe. During which process do facultative anaerobes require more sugar to produce the same amount of ATP? during fermentation. So if you are have ever run a marathon, you'll notice that a marathon is about 26.2 miles, and you'll notice a lot of people really start dragging around mile 20 to 24, depending on how much they've trained. And that's because their bodies are starting to run out of glucose. Number one, their bodies are starting to run out of oxygen, regardless of how hard they're breathing, and so their bodies tend to convert to that anaerobic respiration, and they're only getting 2 ATP out of every molecule of glucose instead of the 32. So hence, that's why you feel very tired. Okay, so let's talk about fermentation, or anaerobic versus aerobic respiration. Why do most organisms use both? Fermentation, remember, is a much faster process. We just talked about it very quickly compared to the lengthy amount of time that we talked about aerobic respiration. So it is good for short-term energy needs. But like I said in the last slide, it only gives you 2 ATP per glucose. Aerobic respiration, on the other hand, yields 32 ATP for every molecule of glucose, and so it can keep up with long-term energy needs. But the process itself takes longer to complete. And it needs a large supply of oxygen. So let's talk about anaerobic respiration. It does not require oxygen. It uses an electron transport chain. When I'm talking about this anaerobic respiration, I'm talking about the anaerobic electron transport. I'm not talking about fermentation. I'm talking about anaerobic electron transport. Remember, this is the one that is just used in bacteria. So, as the name implies, oxygen is not the final electron acceptor because this is an anaerobic process. And oftentimes the final electron acceptor is like a sulfate. And so it ultimately produces H2S, or hydrogen sulfide. And this is used by a lot of bacteria in marshes. And this is why you get that rotten egg smell in a lot of marshes. And this process also starts with glycolysis in the cytoplasm. And again, the yield here is only 2 ATP. So, continuing our review of anaerobic versus aerobic, which one particular step of cellular respiration did we emphasize as anaerobic? That's glycolysis. And which part of cellular respiration is aerobic? The electron transport chain. And remember, that happens on the inner membranes of the mitochondria. So at the end of your chapter, it has a little bit about the evolution connection and the importance of oxygen. Both aerobic and anaerobic respiration start with glycolysis, and glycolysis is therefore considered the universal energy harvesting process of life because everybody does it. Everybody doesn't do it the same. Well, it is the same process, but other organisms have added on to it. So we think between 3.5 and 2.7 billion years ago, before there were high oxygen levels in the Earth's atmosphere, that the first prokaryotic cells used glycolysis to make ATP, and that is the only way they got energy. They just used glycolysis. For every molecule of glucose, they got 2 ATP, and that was it. So glycolysis occurs in almost all organisms, suggesting that it evolved very early and is common to all the domains of life. And so here's kind of a timeline of oxygen with the presence of life on Earth. And you'll notice that the origin of the Earth was about 4.7 billion years ago. Prokaryotes showed up about 3.5 billion years. So for over a billion years, it was just the prokaryotes. Okay, oxygen did not increase in the atmosphere until about 2.7 billion years, and it didn't even reach 10% until maybe 2.2, 2.3 billion years ago. And then it was shortly after that that you have eukaryotic organisms showing up, and they took glycolysis a bit further and started their aerobic respiration and became much more efficient. So let's do a review of the entire chapter. What is cellular respiration? It's a process that converts chemical energy in the form of glucose stored in the bonds of food molecules into ATP, and that ATP is then used by the cells. Where does cellular respiration take place? Overall in the mitochondria, but also in the cytosol, where glycolysis takes place. What are the overall beginning components of cellular respiration? Sugar and oxygen. What are the end products of cellular respiration? Carbon dioxide, water, and ATP. Why is cellular respiration important? It provides organisms with energy from the food they eat. What are the 4 steps of cellular respiration? We start out, and this is aerobic, start out with glycolysis, pyruvate oxidation, then the citric acid cycle, and then the electron transport chain and chemiosmosis. And remember, the electron transport chain and chemiosmosis together are sometimes referred to as oxidative phosphorylation. What do anaerobic and aerobic respiration mean? Anaerobic is without oxygen, and aerobic is with oxygen. Which steps are anaerobic? Glycolysis is the anaerobic step. The aerobic step is the electron transport chain. What are the 3 types of organisms in relation to their oxygen use? Aerobic, anaerobic, and facultative anaerobe. And remember, facultative anaerobes can do aerobic, but as needed, they can do anaerobic. Which form of respiration is the oldest? Glycolysis. Why do organisms use both anaerobic and aerobic respiration? Anaerobic is good for short bursts of energy, and aerobic respiration is good for long duration energy and provides more energy. Remember, anaerobic, you get 2 ATP for every molecule of glucose. Aerobic, you get 32 ATP for every molecule of glucose. So let's continue with the review. Which macromolecules are broken down for energy? This is a good review question. Carbohydrates, proteins, and lipids. Remember, what are those called? Those are the molecules of life. And do you remember the 4th molecule of life? Remember, that was nucleic acids. And we do consume nucleic acids. We eat the DNA contained inside the strawberry cells. When we consume a strawberry, we're eating the strawberry cells, and that contains DNA, but it's really not in a high enough quantity to qualify as a number of calories. Do these molecules all use the same general pathway? Yes, they do. However, remember that they may enter the pathway at different points along the way. Finally, how do photosynthesis and respiration relate to each other? Again, I hope you take the two equations and put them side by side and study them, and you'll know that one is the other one flipped, and therefore photosynthesis uses the end products of respiration to make the beginning components of cellular respiration. On the other hand, cellular respiration uses the end products of photosynthesis to provide energy and make the beginning components of photosynthesis. So it's a cyclical process. I say that Mother Nature is the ultimate recycler. So you may be feeling like I feel at the end of this, that this is a lot of information. And like I said, you can never spend too much time with this information. It takes multiple exposures for this to sink in. There are two worksheets for you, one worksheet on photosynthesis and one worksheet on respiration. Those are posted on Canvas, and so you can take a look at those. And as usual, if you have any questions, don't hesitate to send me an email, and if necessary, we can set up a conference and go over things that way if you would prefer. Have a great day.